Okay, let's go ahead and begin. On the calendar today, it says Intro to Genetics and DNA Replication. So, let's go ahead and do it. Now, in Unit 5, you guys did Mendelian genetics. It involved Punnett squares, dihybrid crosses, pedigrees. You had terms like codominant, incomplete dominant, complete dominant, wild type, mutant type, homozygous, heterozygous, multiple alleles, polygenic inheritance, sex-linked traits, so on and so on and so on. This is also genetics, but it's not going to be Mendelian with about inheritance patterns. It's going to be the actual biology, like the molecules. You got DNA, RNA, proteins, transcription, translation, semi-conservative replication, all those big, big words. And it's going to be enzymes and proteins and molecules. This is the most vocab-heavy unit of the year. So every day while we're working, I'm going to try to do my best to indicate that vocab for you by highlighting it to you, because you really need to speak the language to understand the concepts. So let's go over a little bit, just a little bit of history. I don't want to get too much into social studies lesson when it comes to this, but I do want to introduce just a few people to you because their findings were pretty important. The first one is going to be Rosalind Franklin. Rosalind Franklin is credited for creating an image of DNA, a double helix DNA to be exact, using X-ray crystallography. This was the early 1950s, by the way. So it's not even 100 years old, our full understanding of DNA. It's still a relatively new science. Now, there's a couple other gentlemen that are kind of, what they did to Rosalind Franklin is very nice professionally. Francis Crick and James Watson. You hear these two guys all the time, Watson and Crick, Crick and Watson. What they did is that they saw Rosalind Franklin's X-ray, and they used that to create a model of DNA. It was like a 6-foot tall model of just wires and pieces of metal, and they were making the double helix. It was the first time anyone has actually seen a model of DNA, but they did Rosalind Franklin dirty in that they used her X-ray and they took credit for their findings, but they would not have ever made that double helix model if they hadn't seen her picture. And as I said, whenever we talk about discovery of DNA, a lot of people would go straight to Watson and Crick, Watson and Crick. They would not have been able to do what they did if it wasn't for Rosalind Franklin's work. So using Franklin's X-ray, they created the first model of a DNA double helix. That was they published their work in 1953. Now, the X-ray that Rosalind Franklin actually came up with looked a little like this. It was, this is early 50s, so it wasn't the best. But that's kind of what it looked like. And if you look closely, you can actually see the double helix. So that's the X-ray that she produced. And based on that, Watson and Crick were able to generate this big, I think it was maybe 9-foot tall model of DNA. All right. Chemical composition of DNA. DNA is a polymer. So Hartley, what are polymers made of? Monomers is right. And Reagan, when we're talking about monomers, exactly what is the monomer of DNA? Nucleic acids is right. No, actually, whoa, back up.
No, sorry.
Sometimes I don't even. I assume you guys are right right after about nucleotides is correct. It's safe. They're composed of monomers called nucleotides. Now, then my next question typically gets students a little tongue-tied. Believe me, guys, I know what I'm doing. I want you all to trust the process this semester. I know what I'm doing. What are the, if a nucleotide is a monomer, what's a polymer? Right? What's another type of polymer? Right. Oftentimes I ask, okay, everybody knows nucleotides are monomers. They say, okay, what's polymer? And then I'll get the answer, nucleic acids? So no, what kind of, like what? What's an example of a nucleic acid? And then it's crickets. DNA is an example. RNA is an example. ATP is an example. Now, let's go talk about nucleotides. Nucleotides are composed of three things. Dan, do you know what one part of a nucleotide is?
A pentose sugar.
Very good. Okay, a pentose sugar. Now, today we're only talking about DNA, all right? So what is the sugar in DNA? Very good. Ryan's on fire. Deoxyribose. Notice it ends in OSE. Those mean sugar. Okay, Dan's got that. What else? What else is in there besides the pentose? Teresa? You got it. I'm going to say that for the third one. I'll put it down here. Nitrogenous bases. In DNA, there are 4 bases. You can have adenine, cytosine, guanine, and thymine. Those are the only parts that are different when you talk about a nucleotide for DNA. And what's the one in the middle here? What are we missing? Pentose sugar, nitrogenous base, third part. A phosphate group. Phosphate is PO4 with a -3 charge. In case you forgot that from chem. Okay, well, to continue with this whole idea of nitrogenous bases, that's what I want to talk about. There are 2 classes of nitrogenous bases. You have purines, you have pyrimidines. Now, a couple ways to help you remember which one is which and which bases belong to which class. So purine, P-U-R-I-N-E. It's only 6 letters, 7 if you have if it's plural. It's a short word when you compare it to pyrimidine. A purine, it's a short word, but it's a big molecule. It's a double-ringed molecule. You have a cyclopentane bonded to a cyclohexane. The door is open.
All right, is it fine if we just go through? We need to be in this.
Go ahead. It's a double ring molecule. Pyrimidines are single ring molecules. So it's a big name, small molecule. And purines are small names, big molecules. The two purines are adenine and guanine. And the pyrimidines are cytosine and thymine. Now, a way that may perhaps help you remember this is, what is the second letter in pyrimidine? Is Y. You also have that same letter in cytosine and thymine. So maybe that can help you remember them. Pyrimidine is cytosine and thymine. Also, actually, I was going to say something, I'm going to say it in a minute. Now, as you all should know, A is bonded to T and G is bonded to C. So you always have a purine and you have a pyrimidine. So my next question is, why a purine and a pyrimidine? Why is it not a purine and a purine or a pyrimidine and a pyrimidine? So if we actually look at the double helix, imagine that we have a double helix of DNA and we flatten it out in a two-dimensional perspective. If you were to do a purine and a purine, it's too wide. So purine and purine wouldn't work because the two nucleotides are too big. So what about a pyrimidine and a pyrimidine? It's too narrow. That can't work. But if you have a purine and a pyrimidine together, it's just right. And that actually does match what was on that X-ray crystallography too. So two purines is too wide, two pyrimidines is too narrow. A purine and a pyrimidine is just right. It's kind of like Goldilocks. All right. Here's another dead guy I'm going to introduce you to. Oop. Chargas Rule. Erwin Chargas noticed something when it came to the ratio of nitrogenous bases. Erwin Chargas realized A is always bonded to T, and C is always bonded to G. And if you're wondering, why do I have 2 lines between A and T and 3 lines between A and G? Those lines are hydrogen bonds. There are 2 H bonds, or hydrogen bonds, that connect adenine to thymine, and there are 3 hydrogen bonds, which you can also call H bonds, that connect cytosine to guanine. hydrogen bonds are pretty easy to break. All you do is you just hydrolyze them. You add water to them and they break. Are there any questions so far? All right. So let's go to the double helix nature of DNA. So we all know that DNA looks like this. You have two strands connected by nitrogenous bases in the middle. The sides are what we call just the backbone of DNA. They're made up of alternating sequences of pentose and phosphate, pentose and phosphate, pentose and phosphate. The middle, if this were a ladder, the part where you put your feet, those are going to be the two nitrogenous bases per step or per rung. Some people like to call it a rung. So Watson and Crick realized that each side of a double helix was a complement to the other. So for example, if you had a strand that had A, T, A, C, G, T, the other strand is going to have the complement T, A, T, G, C, A. So this goes back to Chargas Rule. I told you first off, he realized that adenine is bonded to thymine and cytosine is bonded to guanine. That's a no-brainer. So what's the word I want to use? So return to Chargas Rule. He has principle number 2. The percent of A is equal to the percent of T, and the percent of C is equal to the percent of G. Now I have a little problem I can give you, I've observed on AP exams. It's a pretty simple problem. You discover a sample of DNA and realize it's double helixed and 30% cytosine. Here's your question. What percent is thymine? Well, you all should know that percentage is equal 100%. If there, according to Chargas Rule, in double-stranded DNA, if there's 30% cytosine, that means that there's 30% guanine. Period. 30% plus 30% equals 60%. 100% minus 60% equals 40%. That 40% is A and T. Can you divide 40 evenly? Sure you can. 40 divided by 2 is 20. So 20% is adenine, 20% is thymine. And there's your answer. What percent is thymine? 20%. Here's another question you might have thrown at you. I've seen this one before too. You discover a segment of DNA and determine 70% is cytosine. 70. Here's my question. Is the DNA double or single stranded? So if you go with this logic, if it's 70% cytosine, then that means there should be 70% guanine. 70 plus 70 is 140. That doesn't make sense. Well, the only reason that in Chargas Rule that the percentage of A is to T and percentage of G is to C is if it's double stranded, because you have to have a partner, you have to have a complement. The answer to this would be it is single stranded. You don't have to worry about the complement because it's not there. So you don't have to worry about percentages equaling each other. So Chargas Rule of percentages is only applicable towards a double-stranded DNA helix. So Chargraph has two rules. One, A to T, C to G. Easy. Second rule, percentage of A is equal to percentage of T, and percentage of C is equal to the percentage of G. Wow, this class goes by fast. Okay. Now, let's get into how DNA replicates. This is definitely going to take us into tomorrow. DNA replication. All right, so here's what I'm going to do. I'm going to go ahead and throw all the vocab at you now. I'm not going to give you the definitions, but I just want you to see that there is a lot of vocab. So just on the topic of replication, which is I would say about 10% of this whole unit, here's all the terms that you are going to see. Conservative replication. Semi-conservative replication. Dispersive replication. Template strand. Complement strand. Leading strand. Lagging strand. DNA helicase. RNA primase. Single strand binding protein. DNA polymerase 3. DNA polymerase 1. Ligase. Okazaki fragment. Named after a Japanese scientist that discovered them. Let's see, what could I be missing? Ooh. Replication bubble, replication fork. Let's see. 5 prime, 3 prime. Might be it. Yep. So those are going to be terms that you're going to be hearing starting now. I'll see some pens moving, so I'll just wait for you. A lot of terms. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, about 18. There's a lot of terminology you need to realize and be able to use. So let's just take them one word at a time. We're going to work on the first three till the end of class today. Conservative replication, semi-conservative replication, and dispersive replication. That's all the time. That's all that I really have time for today. So this is going to get us to the three models of DNA replication. So when scientists were really starting to learn about the intricacies of DNA replication, we obviously know that DNA replicates during S phase. So that was a known. Why does this happen? So each daughter cell has DNA. You can't have a cell that doesn't have any genetic material. Every cell must have DNA so that it has instructions on how to behave and operate. Without those instructions, it's just a waste of atoms and molecules. So scientists formulated 3 ideas as to how DNA could possibly replicate. And that's what I want to focus on for the rest of class today. So the first idea is going to be called conservative replication. Now focus on what the word conservative means. It means to save. Like, I'm going to conserve my energy for later. I'm not going to waste my energy now or use my energy now. In conservative replication, the original, which we call the template strand, is kept to produce new daughter strands. Now, I want you guys to keep in mind, I'm going to show you 3 models. One of them is right. These are the ideas. These are the proposals on how DNA replicates. I will tell you which one is right after we go through all 3. So as I go over conservative replication, some of you might know that this is or is not the way DNA replicates, but that's not the point. We just want to see how they actually formulate it. So we're going to start off with our parent strand. And the parent strand just looks like a regular segment of DNA. But then we're going to have our first replication. And the navy blue is going to represent the parent strand, everybody. The sky blue is going to represent the complement strands or the daughter strands. So here we go. So you keep the parent strand, it becomes one of the 2 new strands. And you basically made an entire copy by using that. And then we'll go to our second replication. So you can follow where the original strand came from and how it moved on from stage to stage of replication to replication to replication. The next idea is called semi-conservative replication. I may not have time to go over the third one, but I should be able to squeeze this one in. In semi-conservative replication, the parent strands are separated and serve as templates to produce daughter strands. Remember, the parents are called templates, the daughters are called complements. The complement is based on the template. The complement is based on the template. So here we go. So there's our parent strand there. I'm just going to separate these two so we don't get them blended in. All right, you can see how on the first replication you kept half, and with that half you made a complementary other half, a complement strand. That's why this is called semi-conservative. Semi means half. You save half to make the other half. So you can follow the path of the original strand all the way from one stage of replication to the next. That's why this is called semi-conservative. And I don't think I'm going to have time for dispersive, but basically in dispersive, you chop up the parent and the voids will fill in to make the complement. So I can at least write in what dispersive means. I don't have time to draw it. So dispersive replication is a molecule is chopped up. I'll just save it for tomorrow. So we'll start that tomorrow. All right.